Yeah, I think measuring degrading enzymes would be a good way of seeing how efficient the microbiota is. Yes, so I would say it's the definite yes for the first question, because of course you need the enzymes there to degrade. But as I showed you also, it's often the whole three-dimensional structure that is important. But of course, we're doing that essentially if we're incubating our microbiota or specific bacteria with these recalcitrant substrates, and you see a breakdown in the substrate is a more direct way of seeing this. Well, the SCFA profile is much more simple than the microbiota profile. So I think, if the SCFA profile tells you something about whether a person is likely to have a healthy microbiota and a healthy metabolite profile. But you can't really go back from a very simple profile to the complexity in the microbiota. So I think it wouldn't be a good indicator for microbiota. I would go for molecular techniques for that.
I think the other question is about the individual genetic variance for human, but for animal production, uniform production is one important issue.
Oh, it just disappeared. Oh, it's on the top now.
What's your perspective for that?
Well, that's a really tricky one. I think the interplay between the human genetics and the microbiota composition isn't really fully understood yet. So I don't think I can give a clear answer to this. There are definitely connections. Some people associate even different blood groups with different types of microbiota. It's also not really clear how individual specific the colonization is. On the other hand, we have all the fecal transplantation work now, and it does look like that some people respond very well to fecal transplantation. This is more done currently to eradicate Clostridium difficile infections. But I think in some people, the new microbiota establishes quite well. In some people, there's a mix between new and the old. So I think it's too early to really say how much the host genetics influence this.
The other question I would like to see, that's from Joran. Can this be achieved through exogenous enzymes as well? I think this question also relates to, for plant fiber, it's very complicated. The structure, particularly the crystalline structure, that actually is the bottleneck. So for the animal production, for utilization of those polysaccharides. So I think this question is also important. Can you address that?
Okay, so it's fiber degrading enzymes, you mean? Yeah. So I think there is a pro and con there as well. Of course, if you degrade them down to the monosaccharides, they will not be a food source for the microbes anymore because the animal will take most of that in. So from a microbiota and short-chain fatty acid producing point of view, I don't think it would be good to completely degrade the fiber. But to make it maybe more accessible, maybe a good thing. But on the other hand, at least from a human perspective, I think you don't want this really totally soluble, rapidly fermentable fiber either. Because then you get a huge activity in the proximal colon. People get gut symptoms, but also then very quickly it runs out because it's a very buffet for everybody. So all bacteria can chip in, or most bacteria can chip in right away, huge activity in the proximal colon, and then very little happening in the distal colon. So I think you want a mix of hard digestible stuff that genuinely takes longer to digest, and you need a longer chain of bacteria to nibble away and slowly trickle it into the system, that you have fermentation still further down. And that probably would help to minimize the risk of gut diseases further down.
I think we still have some time for questions. The coffee starts at 11 o'clock, correct?
Yeah.
Yeah, if we probably can go through some questions again. I think, in human, the main intervention through the diet to boost the endogenous production of a build rate. For you, what is the best way to go in terms of a diet composition?
Okay, what's the best diet to have high bitrate production? I guess that's how I interpret the question. I think it is individual to a degree, but you remember my in vitro data, which does show that certain fibers are more stimulating to overall butyrate production or overall propionate production. So I think fructans are a good source for butyrate, at least in the human system, and starches are also good. Ruminococcus bromii, by the way, who I showed you as the main keystone species for starch degradation, does not produce butyrate itself. So the butyrate production is not by direct production from Ruminococcus bromii. This is cross-feeding effects. But the Ruminococcus bromii actually grows quite poorly. In the lab, we're also having trouble to grow it. It needs a lot of vitamins, so it also is normally not there in very high amounts in people. I think we boosted it so much in our human study because we gave the system so much starch. So probably even in the gut, it's actually adapted to be a slow grower. It doesn't take all that much of the starch for itself, I believe, or I assume. A lot of the starch it makes available to other bacteria. And then you have butyrate-producing bacteria that can then turn the breakdown products of the recalcitrant starch into butyrate. But to a degree, it may, again, be an individual difference in different people. So I think we could design diets that will generate high butyrate in most people, but we need to be aware that in some people it could be different. This has also been shown for lactate many years ago, where there were in vitro incubations with lactate, and I've shown you that some bacteria turn lactate into butyrate and others into propionate. And I think there were 3 people in the study, and 2 were very butyrogenic, and 1 person almost converted all the lactate into propionate. So there definitely can be differences between different people. It's not necessarily a one-size-fits-all.
I think the other question is a pretty simple question. Do you think different species have similar microbiota? That should be pretty straight question to answer.
Yeah, I think mostly we would say if you find the same species in different animals, they tend to be quite similar. So even I showed you the cellulose degraders, which are mainly present in the rumen because their cellulose degradation, the plant cell wall cellulose, is so important for feeding the rumen animal itself. So this bacterium Ruminococcus flavifaciens has this cellulosome, this enzyme apparatus to degrade cellulose. And the Ruminococcus champignonensis I showed you from the human gut is a close cousin. It's a very related species in the same genus, and it seems to have a very similar role in the human gut. So yes, I think again, it's biology, it's nature, it's messy. I can't really give a blanket, they're all going to be the same, but mostly we find the same species in different animals has quite similar functions.
So the next question is the activity of the starch and fiber degrading enzyme would be good indicator for the good microbiota.
Yes, I would say so. I can't see the question right now. Yeah, okay.
Okay. Do we have more questions? Good indicator.
Yeah, I think there is a link between pH and health. So again, I'm not an expert on the physiological side of things, but I think the lowering of the pH is stimulating butyrate producers. So I think what we find in vitro in our fermenter studies and also in our batch cultures, if the pH is around over 6, the Bacteroides seem to be more fit. The Bacteroides is winning out. The butyrate the Firmicutes is hanging in there, but they're not growing as fast. So I think that the pH goes down really means the Firmicutes have more of a chance. And don't get me wrong, it's not all Bacteroides is this and all the Firmicutes are that. Within the Firmicutes, we have some that are more pH tolerant and less. You may remember one of my slides with the 2 Rosburias, which were different. But I think if you want good butyrate production, lowering of the pH is important. Otherwise, the butyrate producers will just not be able to compete that well to the Bacteroides, which are propionate producers. But on the other hand, it can spiral out of control. In ruminants, there's something known as lactic acidosis, which you may be aware of. If the system spirals out of control and the pH drops below a certain point, if it gets sort of below or into the below 5 region, then the whole system can spiral out of control because most bacteria can't grow anymore and a few are taking over now. So even in ulcerative colitis patients, you find some patients in severe colitis which have a lot of bifidobacteria. And of course, everybody thinks bifidobacteria are really health promoting and really wonderful. But actually, if they're taking over and suddenly there's only bifidobacteria, it's not good anymore because they're lactate producers. And if the butyrate producers can't go for it anymore, then that's not a good thing. And they're probably then a sign for the system has gone out of control. The bifidobacteria are just more hardy. They can withstand the low pH better. So yes, pH lowering is important, but not too much. So again, if you give too much substrate for the bacteria in one go, there's a higher risk of this going out of control.
There are a couple more questions. I think two of them Petra has answered already, similarly, questions. And I think the last question would be keto diet. So do you expect, if we are on keto diet, do you expect your immune to fall down? And how do you propose
Don't continue on the keto diet. I mean, just don't do it. I think it's just no need. There are better ways to lose weight and be healthy. I just don't even want to think of a way to now counteract the bad effects of a keto diet. It's just a bad idea.
Or take more butyrate. Take more butyrate. Butyrate can reduce the lipid production.
Yeah, but also, fiber is important for many things. It's not just the short chain fatty acids. Fiber is also important for water holding and transit time. And
Can I ask two simple questions? You mentioned phytochemicals also affect the microbiota. I just wondering in what kind of way approach to affect.
It's very complex. So, you can see this in many different ways. Some phytochemicals may inhibit certain bacteria. They can be toxic to a bacteria. So you potentially change the profile in the microbiota. Of course, people want to believe that phytochemicals are bad for proteobacteria, which we don't want to have so much of, but are leaving other bacteria alone. It's not going to be that simple, but you can have an inhibitory effect. But more importantly, the microbiota is very closely involved in the biotransformation of many phytochemicals. So a lot of the phytochemicals are covalently linked to the fiber, for example, as I showed you, and other phytochemicals are soluble in the vacuole of the plant, but they're linked to sugars. So if they're glycosylated, then basically we can't take them up. And then also only when there's glucose, we have some enzymes that can cleave these sugars off, but a lot of them cannot be digested by ourselves. We need the microbiota to liberate these phytochemicals, the aglycones, the actual polyphenols, to either then have their action or to be absorbed. A lot of them can only be absorbed once they have been deglycosylated by the microbiota. And then even the aglycone is further transformed, dehydroxylated, dehydrogenated, or hydrogenated by different bacteria, and then their bioactivity may change. So some of these phytochemicals only have their health beneficial action once they have been further metabolized by the microbiota.
Okay, my second technical question. You talk about in vitro subculturing because you do the modeling like using the in vitro culturing. What kind of a medium do you use? Because that's very critical. The selection of medium will select your result and also conclusion.
Yes, it's very crucial, and there is no one medium that is perfect for all. But the route really comes from a rumen background. So when we moved into human research about 20 years ago, we used the media that were used for rumen bacterial growth. So we use a lot this medium M2, which has rumen fluid in it.
Okay.
And if you compare in a human fecal incubation, if you compare this medium over BHI, which is used by many brain and heart infusion, you get roughly 10 times the number of bacteria. So it's much better. But for our Danone work, for example, with the vitamin, we had to go to the other extreme. We had to take everything out. There could be no yeast extract, not even normal cas amino acids, and it's getting very hard to grow some of these bacteria in that. So.
Okay, thank you so much. And Petra got lots of questions.
Thank you.